Good morning, good afternoon, or good evening, depending on where you are in the world, and welcome to today's webinar, which is sponsored by Cambrex. Before we get started, just some brief administrative things. First, we are recording the session and we'll share this with you later along with a PDF of the slides. Please note that sometimes the email with the PDF sometimes goes into spam, so please have a look out in that folder. We also put links to the webinar archive on our website. Secondly, any audio problems, please do contact Claire in the chat area and she will try to help. Sometimes simply logging off and back in again works well. Finally, if you have any questions for our speaker, please type these into the question area of your dashboard and I will relay these to Adrian at the end of the presentation as time permits. And with the ones we don't get to in the hour, we'll follow up offline with those. So now it's my pleasure to introduce you to our speaker. Dr. Adrian Amador is the Director of Process Chemistry at Snapdragon Chemistry, a Cambrex company, where he leads a team of 12 process chemists developing efficient manufacturing processes for the pharmaceutical, specialty, and agrochemical industries. He joined Snapdragon in 2018, and before that he began his research career studying photochemistry and spectroscopy at the Georgia Institute of Technology before completing a PhD in chemistry at the University of Wisconsin-Madison, focused on novel photocatalytic methodologies. He then carried out postdoctoral research at the University of Minnesota, working on sustainable polymers. His current research interests include the use of automation, data-rich experimentation, and data science to improve process development and enable more robust early-phase manufacturing, with a particular focus on complex synthetic therapeutics such as peptides, oligonucleotides, and synthetic polymers. He's now going to show some of his work with this presentation entitled Supporting the Development of Scalable Manufacturing Processes for Complex Synthetics Through Automation and Machine Learning.
Great. Well, thank you very much, Ben. I'm so happy to be able to give this presentation today. And so as mentioned by Ben, I'll be discussing some of our approaches to utilize technologies such as automation, machine learning, and other types of algorithmic, data-rich processing approaches to support these complex synthetics therapeutics that have become more and more prominent in the pharmaceutical industry. So first, a brief introduction on Snapdragon and Cambrex as a whole. Cambrex is a multinational CDMO with sites located throughout the United States and Europe, including several large manufacturing sites, as well as some smaller early phase and development sites. Much of the work that I'll be discussing today is coming out of our Boston site, which is considered our center of excellence for process development and early phase development in complex synthetics. So a brief outline before I jump into the meat of the talk. First, I'll go into a brief introduction on opportunities for complex synthetics process development, and then I'll go through a series of case studies. First is software tools and automation for predictions and physicochemical characterization, mostly around the solubility predictions for polypeptides. Then I'll go into a couple of case studies around automated workflows for process optimization. Both of these are, two of these are closed loop optimization systems, one using a more standard snob fit approach, which is stable noisy optimization by branch and fit for optimizing CBZ deprotections for peptides in the liquid phase. The next is a more advanced optimization approach using Bayesian optimization for the optimization of a solid phase oligonucleotide synthesis process. And then I'll dive into some of our recent developments on developing a automated liquid phase peptide synthesis platform to further facilitate the development of these types of manufacturing processes. And then finally, I'll close out this webinar diving into some automated workflows for analytical development, particularly around the use of Bayesian optimization for rapid reverse phase HPLC method development. And so some of the reasons why we're looking at using some of these advanced technologies for complex synthetics and synthetic drug substances is around some of the trends that we're seeing in this industry overall. First of all, there's a increasing level of drug complexity. This is very obvious with the oligonucleotides and peptides. This requires improved and automated workflows for analytical and QC development, as well as increased automation and precision control in manufacturing. There's also increasing time pressures for developing these drugs, which is requiring more standardized and automated process development workflows, as well as the use of platforms and AI-powered analytical method development. And there's further increased scrutiny on the sustainability and the security of supply chains. And one of the approaches for tackling this is around the use of solution phase processes for these complex synthetics, both the peptides and the oligos. So overall, these trends benefit from the implementation of advanced technologies such as automation and machine learning based approaches. And so more and more over the last few years, Snapdragon and Cambrex has become more involved in the complex synthetic modalities. This includes some of the more non-traditional small molecules, monodispersed biomacromolecules, as well as the polydispersed macromolecules. So most of today's talk will focus on the center column here, the monodispersed biomacromolecules such as peptides, oligonucleotides, and similar types of modalities. And really focusing in on the use of automated development workflows as a supporting technology to enable process development in these areas. So the complex synthetics market, which includes the oligos and the polypeptides, is clearly a steadily growing sector within the pharmaceutical industry. Both the peptides and the oligonucleotides are expected to have a substantial growth over the next 10 years. However, these modalities, they present increased complexity both on the process side and the analytical development side, which leads to higher production costs for these types of therapeutics, greater difficulties during process development, as well as more complex analytical characterization due to the number of discrete impurities that are expected to be formed during the manufacturing process. So overall, we think that algorithmic optimization is likely to serve as a highly effective approach to enhance process development efficiency for these modalities, as well as analytical development. So Snapdragon Chemistry was originally founded in 2015, and we've put a large focus both on technology development as well as the contract development areas of our business. And really all of these things, both the contract development for process development, analytical development, and technology development feed forward into manufacturing. So we try to make use of more advanced types of hardware, our own internally developed softwares, as well as substantial use of automation to enable more rapid entrance into manufacturing, as well as more robust GMP manufacturing of complex APIs. So for the first section of this webinar, I'm going to briefly touch on some of the software tools that we use for solubility predictions. So having early knowledge of a peptide's solubility can be highly invaluable to facilitate process development. We see this most heavily for the peptides currently in our own work. With the increased production demands for peptides, there's a very large focus on the use of liquid phase peptide synthesis to manufacture these types of molecules. And the type of approach that's most often used for LPPS is a fragment-based approach, where you'll have your full-length, longer peptide. A lot of the more recently approved drugs are in the kind of the 30 to 45 mer range for peptides. But this would be broken down into individual fragments that could be made either through solid phase or a fully liquid phase approach to make these individual fragments. And then they're combined in the solution phase to make the full-length peptide. And so having a good idea on the individual solubility of these fragments is quite beneficial, as you don't want to be making fragments that will not be soluble and thus difficult to do the solution phase conjugation or even just make in a fully LPPS process. And so that factors into the appropriate selection of your individual fragments. And so having the relative solubility of these potential fragments is one of the most critical considerations when you're going into fragment selection. So there's been several approaches reported in the literature over the last few decades for predicting the relative solubility of side chain protected peptides, just based off the peptide sequence. And so this enables a simple sequence to expected general solubility prediction to serve as an enabling tool. And it can be used to evaluate the relative solubility of candidate fragments. So these predictions are typically based on models of trained on large data sets, typically from solid phase peptide synthesis. So one early reported approach that we utilize heavily at Snapdragon is built around the swelling volume that's observed during SPPS. And this can be correlated to the relative degree of peptide aggregation. It's basically an inverse correlation where the more aggregated a peptide is, the lower the swelling volume that's observed. So by analyzing the relative resin bed volume during SPPS of several hundred peptides, a predictive aggregation potential model can be built based off of that peptide sequence. So this can then be turned into a simple Excel-based tool for ease of use for prediction of peptide solubility. And we've actually found this to be highly predictive, even for liquid phase peptide synthesis, despite the fact that solid phase peptide synthesis data was used to develop this model. We find that this actually tracks quite well when we go into liquid phase for understanding when a peptide might precipitate out of solution. Now, there are more advanced approaches that are used and reported more recently in the literature. On this slide, I'll share one example out of the pentalute group where they've used UV-vis data during Fmoc deprotection to have an understanding of when peptide aggregation is occurring. And so then you can correlate this data to structural representations of individual amino acids to then develop a similar predictive model for sequence to aggregation and solubility issues. So moving into the future, something that we're currently developing using data from high throughput solubility workflows as well as high throughput crystallinity workflows is to have more granular predictions around this. By measuring the solubility of a large library of peptides, we'll be able to build sequence-based prediction of individual solvent solubilities for different peptides, as well as have a prediction of the likelihood of isolating crystalline intermediates for peptides. So moving into the next section, largely centered around algorithm-driven auto-optimization of reaction conditions. This can be a bit more of an involved development area, and it requires a few critical pieces coming together for this to work effectively. So the early work that we've done in this area is around the auto-optimization of flow reactors. And so you need a robust flow reactor platform. You need to be able to have some sort of online analytics to read out either the purity, yield, or throughput of your process. And then you need an optimization algorithm that can take in the analytical data and provide reaction conditions to be testing to gradually work towards your more optimal reaction conditions. And so around this, we've built flexible automation to easily incorporate new hardware as well as different types of PATN sensors. We've built robust controls so that we have good safety interlocks, have remote access, it can detect issues within that. And we've implemented multiple different types of algorithms, both SNOBFit, which is typically only used for continuous variables, and simpler design spaces, as well as Bayesian optimizers, which is quite effective for multi-objective optimization and a bit more effective for complex design spaces. And so we've utilized a lot of our own internally developed platforms, as well as collaborated with other industry providers to do this type of work. We have our own modular flow platforms to do the process side of things. We've collaborated with Mettler Toledo to use their Easy Sampler to collect samples from our reaction platforms, developed our own sample relay system to then take these samples and deliver them directly to an HPLC, such as a Shimadzu or Agilent HPLC, and then utilize some form of algorithms to then interpret those results and provide reaction conditions to be testing throughout the system. So one brief case study I'd like to discuss is the optimization of a hydrogenation that's used as a key deprotection step during elongation of a peptide fragment in the liquid phase. And so in this synthesis, we are using some sort of aliphatic tag attached to the C terminus of our peptide, and then deprotecting that using hydrogenation to remove a CBZ protecting group. And then this can enter this repeating peptide elongation cycle where we'll do amino acid coupling and then deprotection and repeat that until we get to the final peptide fragment. So this CBZ deprotection is the reaction of focus for this slide. And so we utilized our combination of pieces of equipment and software in order to quickly optimize this step for each of the intermediates throughout this process. And so we're utilizing a packed bed flow reactor with a liquid stream of our growing peptide chain, hydrogen as a gas, and then being pumped through a palladium on aluminum packed bed and collecting samples at the outlet of the reactor, which can then go into a UPLC and that data input into an optimization algorithm to provide reaction conditions for testing to gradually work towards the optimal reaction conditions. So we're able to use this type of algorithm to rapidly search the design space around this, and we just use temperature, pressure, and hydrogen flow rate as our continuous variables, and we're able to efficiently explore the overall design space to make sure that we're finding the global maximum for purity. And using this type of approach, we're able to optimize each of these deprotection steps within a 6-hour time window to achieve the maximal purity for our peptide after deprotection. So more complicated case study for a more difficult process to optimize that we looked at is solid phase oligonucleotide synthesis. So for each addition of a phosphoramidite, there's a multi-step cycle that's required, consisting of activation of your incoming phosphoramidite and coupling to your growing oligonucleotide chain, oxidation of your phosphorus, capping, and detritylation to prepare it for the next cycle. And another difficulty that's introduced here is the fact that this is being carried out on the solid phase, so this makes rapid analysis of the ongoing process quite difficult. So we wanted to use a more advanced optimization algorithm, such as a Bayesian optimizer, to try to tackle this. And Bayesian optimization is known as a very effective method for optimizing an objective function where the analytical form of your objective function, and by that I mean how the process parameters translate to a specific yield and purity is unknown. So it's essentially a black box. And the experimental cost is quite significant. So each experiment that you need to run takes a lot of time and resources. So you don't want to be running 1,000 reactions to get your optimal experimental conditions. And so just to briefly describe how a Bayesian optimizer works without delving into the math too much, since we don't know the analytical form of our objective function, we use a surrogate model, which is typically a Gaussian process to gradually optimize this surrogate function to have some predictiveness around our design space for how process parameters would then translate to a yield or purity. So you can just see how this surrogate function is optimized as we go from no data to a small amount of data to enough data points to have some level of predictability and reduce the error bars of our prediction. So another piece of this is how we design our objective function. So for SPOS, we wanted to look at a multi-objective optimization rather than just looking at purity or just looking at yield. We wanted to be able to optimize for both, in this case study, both purity and the cost of the overall process. And so there's a few ways that you can fashion your objective function. The simplest way is a weighted sum approach where you have a weight for each of your objectives. So maybe you have like an 80% weight on purity and a 20% weight on cost. And then your overall objective is just a linear combination of those objectives and their weights. A more complicated approach is often termed the chimera objective value, where you might have a hierarchy of objective importance. So you might indicate that your oligonucleotide purity is your highest priority, and then individual impurity levels as your second highest, and then cost is your third highest. And then you can have thresholds to allow some variation that you can sacrifice before you start to look at your other objectives. And so to explore the use of this type of optimizer for SPOS, we designed a custom synthesizer that allowed us to have complete control over their overall system, as well as to connect it with our LabOS software platform for process control. So this was designed very similar to a typical off-the-shelf oligonucleotide synthesizer where we have a series of valves and pumps or a circulation loop. But in this case, we built in a series of columns with a column switching valve to evaluate series of experimental conditions as we go through the optimization. So this is what our kind of custom-built synthesizer looked like. Just use a series of HPLC pumps, switching valves, as well as a series of columns to be able to evaluate conditions relatively rapidly. And so for this multi-objective optimization, we selected a total of 12 variables for optimization. We wanted to look at each of the cycle stages throughout the SPOS process. For each of these stages, we look at the relative flow rate for each of these stages, as well as contact time. And then for the coupling reaction, we also looked at the equivalence of activator, as well as the equivalence of phosphoramidite being used. And then as we're going into this optimization, we have our standard conditions. That's kind of our starting point. We have a lower bound and then an upper bound for each of these variables defined within the optimization. And so just looking at our base case with our initial conditions, this is a relatively small reactor with unoptimized flow paths. So we do have to use a relatively large excess of phosphoramidite, and activator is expressed as the ratio relative to the phosphoramidite. So this would be 5 × 26 as far as the overall equivalence of activator relative to resin loading. So our base case provided a purity of about 60% for a arbitrary 10 mer. 60% overall purity. Just looking at the individual oligonucleotide species that we could define is about 86%. And so this was our starting point going into this optimization. So we tried to approach this using both a weighted sums approach as well as chimera optimization. The initial portion of the optimization utilized weighted sums. And so we just had purity and cost with individual weights to do this. And going through around 55 experiments using weighted sums, we did not see particular progress around optimizing either of those objective values, the purity or the cost. You know, this is kind of quite noisy, bouncing around. So the green is the overall purity and the blue line is the overall cost. But really no obvious trend that either of these is improving as we're going through the optimization. We then switched to the chimera optimizer where we had purity as our highest objective value with a tolerance of about 10%. So this allows a sacrifice of 10% purity if that also increases or decreases the overall cost of the process. And you can easily see that as soon as we switch to the chimera optimization, we're starting to get much better improvement on both costs. These blue lines here, you can see kind of a gradually decreasing trend here, as well as the purity where we're consistently getting higher and higher purities as we go through this optimization. So this initial portion using weighted sums utilized 55 data points for optimization without much progress. And then over the next 48 data points for the chimera optimization, we started to get substantially better improvements to our system. So putting it into a scatter plot, you can easily with this see the improvement trend here. To the left of this red line, we see the weighted sums trials where it's relatively noisy without much of a decreasing trend in costs that we're looking for. And then once we switch to the chimera trials, we start to see a relatively consistent improvement in our cost. And a similar observation was observed with the purity, where it's relatively noisy with the weighted sums, and then we see gradual improvement as we switch to the chimera trials. And so one of the really powerful features of doing an optimization such as this and having all of these data sets is then you can really start to dig into the data and explore this quite effectively. One of the things that we could do using the software platform that we're utilizing for this is define ranges that we wanted to explore to see what other types of conditions are present to still meet those objectives. And so in this case, we're able to define a specific merit value, which is the overall objective value combining both cost and LCAP, and constrain this to the top 10% of merit values, and then see what types of process conditions provide these highest levels of objective values. And so then this gives you a range for all of your different process parameters that you might want to work with. And so, maybe what the algorithm initially decided was the most optimal conditions, in retrospect, ended up not being the most optimal conditions. Maybe we instead want to reduce all of the hold time, so recirculation time, capping time, coupling time, et cetera, so that we can decrease the plant time that we're doing this process. And so we can see within this merit value what types of conditions provide the shortest cycle time overall for this, while still maintaining relatively good purity and cost. So this is the overall improvement that we saw throughout this optimization process. We saw a 22% reduction overall in cost from our standard conditions and 11% increase in the crude timber LCAP over the course of just a couple of weeks of optimization, with minimal manual manipulation of the system being required. So in the next section of this webinar, I'm going to discuss a fully automated LPPS system that we've designed to help accelerate process development of liquid phase peptide synthesis processes. So overall, it's well recognized that liquid phase peptide synthesis presents a substantial advantage in the form of scalability over solid phase peptide synthesis. But unfortunately, LPPS is not a plug-and-play platform compared to SPPS. And so this presents a substantial bottleneck for upfront development. Over the last several decades, SPPS has become a platform technology that's highly automated, and it requires minimal upfront process development cost and time in order to get to a manufacturing-ready process. In contrast, LPPS requires a substantial amount of development and cost to get to a similar state of readiness for manufacturing. So what we want is a faster process development technology to use with LPPS. This is kind of a critical missing piece of the puzzle for wider adoption of LPPS, and not have this substantial development cost that makes it a little bit less attractive than SPPS, particularly for early phase projects. And so what we've wanted to do is try to utilize automation of LPPS in order to kind of remove this bottleneck. And so one of the things that we've observed is that LPPS is going to require some complex automation and recipe programming due to the fact that it's all in the liquid phase. You're not dealing with just a resin bed that you can filter away the unreacted reagents and byproducts from the system. And so you need high fidelity automated liquid-liquid phase separation. So one of the things that we're trying to do is implement advanced technologies such as AI, so that we can have human language to machine automation enabled within the system, as well as have advanced approaches to assist in decision making throughout the process, whether this is in phase cuts or in the overall optimization itself. And so to try to enable this, we have designed and built a fully automated LPPS system that can accommodate both Fmoc protecting group strategies as well as CBZ protecting group strategies, with a main reactor where most of the chemistry and coupling chemistry is occurring. So if this is an Fmoc process, most of the chemistry, both coupling and deprotection, will occur here. If it's CBZ chemistry, the coupling will occur in this main reactor, and then deprotection of your CBZ protecting group will occur in your hydrogenation reactor. And so we've been successful in building this system, and we've already been using this for a large number of projects to try to accelerate process development for peptide fragment synthesis. And so just graphically, the overall process for an LPPS process in our labs is we have a deprotection reaction. This is a homogeneous, generally single phase system Once that's complete, we do liquid-liquid extraction to wash away excess reagents and byproducts. And then this can move forward to, again, a single-phase coupling reaction where we add in the next amino acid, activator, and coupling reagents, and then proceed to the next liquid-liquid extraction. So we're using this for a variety of purposes in our labs. One of these is to have early identification of challenging and problematic reaction steps. We also use it to quickly access materials for testing fragment assembly. We use this to generate materials for measuring the solubility of individual peptide fragments, as well as generate material to screen for isolation points and potentially crystalline intermediates. And so I'm going to show a brief video and talk through this as the process is ongoing. So what we've designed this to be is a fully human intervention-free approach for tag-assisted LPPS, and to use this to eliminate some of the bottlenecks in development. So going into this time-lapse video, first we're showing the amino acid activation step on the left, and then this transfers over to the coupling reactor for the amide coupling reaction. Once that's complete, it then proceeds into a liquid-liquid extraction where we have a series of aqueous washes. You can see a phase cut occurring now, and then this will automatically do the phase cut and proceed into additional aqueous washes to remove excess coupling reagent, excess amino acids, and other types of byproducts. And so this is another phase cut. So we're mixing the two phases. We allow the phases to separate. We're removing oxima activator here. And then this will, after this next phase cut, split off the aqueous phase to remove this from the system. The similar process happens for deprotection. And so we add a scavenger for dibenzofulvene for Fmoc chemistry. The deprotection then proceeds. And then once that's complete, we then proceed to a series of aqueous workups. Again, with automated removal of the aqueous phase, which contains excess base and any amino acid byproducts that are present in the system. So going into the next slide, we've tested this in a large number of different peptide sequences. But one case study that I can share is just utilizing this for the synthesis of one of Tirzepatide's fragments, which has been reported previously in the literature by Eli Lilly. So for this case study, we used a simple allele-based tag that can be removed later by palladium catalysis. We wanted to test the robustness of this system for synthesis of this tag, just using a generic recipe with no prior optimization of this process for this specific sequence. And so within our system, we have a tunable automation recipe for each step, but a chemist or engineer or operator can go in and use our interface that's integrated into the ELN to execute this automated recipe to go through this sequence of amino acid couplings and deprotections to build out the full fragment. So on this slide, I'm just showing the IPC HPLC data as we proceed through the synthesis of this 14-mer, starting from the dipeptide on the top trace here. We have a small molecule excess reagents and impurities as we're going through the synthesis on the left side. So you can see kind of clean transition from the dimer up to the 14-mer as we proceed through this process. And generating this final 14-mer peptide after precipitation in a little over 90% purity without any sort of purification aside from precipitation. And generating this with on around 35 gram scale with orders of magnitude less waste than would be generated for the equivalent SPPS process. So in the final section of this webinar, I'll dive into some algorithm-driven approaches for auto-optimization of analytical methods. So there's some of the key challenges for traditional HPLC development is that it's time-consuming and often a trial and error process. It can be resource and cost-intensive, and there can be challenges for method transfer across labs. And increasingly, particularly in the case of complex synthetics, we're dealing with increasingly complex sample matrices where there's a large number of individual impurities that are expected to be produced throughout the manufacturing process, such as deletions, double additions, and things along those lines. And so what we've developed and implemented is a platform to further accelerate this HPLC development timeline. This starts with an automated screening approach where we screen multiple columns, different column chemistries with acidic, neutral, and basic mobile phases to rapidly explore the separation space. Once we've identified our ideal discrete variables of column chemistry and mobile phase pH, we then move into an algorithm-driven optimization where we can refine the selected screening conditions using a structured design and multi-parameter optimization, often focusing on critical peak pairs to improve resolution and peak shape within the analytical method. And this can be coupled with automated synthesis platforms such as SPPS platforms to rapidly synthesize a library of expected impurities. These typically are standard for SPOS or SPPS. The expected impurities, you'll often see deletions, you'll see double additions, you'll see epimers, particularly in the case of peptide synthesis. And so you can consider these as standard process relevant impurities, and rapid synthesis of these things is quite facile and can be plugged into this automated development workflow. So giving a overall overview of the screening part of this development, we have 3 sets of mobile phases with variable mobile phase pH, and then we test all of these across 10 columns. We look at a range of different types of gradients, so starting point gradients as well as the length of the gradient. And then we've got a desktop application that fully automates this experimental screening and performs integrated data analysis and reporting. So looking at the screening strategy, we selected a wide range of different types of mobile phase chemistries. So C18, C8, and then a variety of other types of stationary phases to be able to tackle a large range of different types of molecules from relatively hydrophobic to highly polar types of compounds. So in this example case study, what was observed for a particular analytical method is that there was multiple shoulders and co-eluting peaks, and we wanted to achieve separation around some of these critical pairs. So in this approach, we first initiate automated screening across the mobile phase pH and the column chemistry, and then we proceed into the algorithm-enabled optimization. And so what this allowed at the end of this process was within just a couple of days, we had a drastic improvement in the overall quality of our analytical method and increased resolution across some critical pairs of impurities. So just looking at the initial column and mobile phase screening, we had a few higher ranked choices for the columns. And in the end, we ended up selecting this combination of a neutral mobile phase and this column number 5 here, which provided just with this initial screening, the best overall resolution. And so from there, we then proceed into the algorithm-driven optimization. And so we go into this with the initial conditions from the screening. The algorithm proposes the next set of parameters. Our software generates the method files automatically and a batch file for the instrument to run. This then gets pushed forward to the HPLC, which then runs the experiments. The chromatograms are analyzed automatically, and the results are then plugged in to the optimization algorithm, where it's decided whether the optimization is done or the next set of experimental parameters are proposed to continue. And so the fixed conditions come from the initial screening, where we have a fixed column. In this case, it was column number 5 and a neutral mobile phase. And then we have variable parameters of the flow rate through the system, the gradient end concentration, the gradient length, and isocratic hold time. And so while there is a large theoretical design space, we only needed to explore about 40 different experimental conditions in order to arrive at optimal HPLC. So this is just kind of driving us through some of the observations as we went through this. And so you can see on this top left, we've only actually got 3 peaks here. And then as we're proceeding through the optimization, we're actually starting to pull out a second impurity from this later eluting peak here. And then finally, at our optimal set of conditions, not only do we maintain resolution between this critical pair here, but we also get complete resolution between these 2 impurities, which in the initial set of conditions just appeared as a single peak. So with that, I will conclude this webinar. So what I hope that I've shown is that a combination of algorithmic prediction tools, algorithmic optimization, and automation can be invaluable for supporting the development of increasingly complex therapeutics. The use of effectively implemented automation can allow for faster LPPS development, SPOS development, access to critical physicochemical data such as solubility and crystallinity, as well as access to critical impurity markers. And then the use of algorithmic tools such as machine learning and other models provides better predictability before you initiate process development, and then once you're in process development, faster optimization around complex design spaces for both process and analytical development. So all of the work that I've discussed in this presentation was carried out by a large group of different chemists, engineers, and analytical chemists. So, you know, a great team of people here at Snapdragon and Cambrex are involved in all of this work and make this possible. So with that, I will close this out, and I'll be happy to take any questions.
Very well, thanks, Adrian. That was a really interesting presentation. We've got 3 questions at the moment, so if anyone has some extra, I think we've got time to answer at least 3 and maybe a few more. So the first question here from Brittany is, for the SPOS example, the first 2 batch sizes are quite large, about 25 batches. And really the question's about whether, you know, how do you select batch size? Would a smaller batch size of being fed back more frequently algorithm actually help with the optimization? And also just really thinking about when did you decide to switch from the initial optimization across to the Chimera objective? And so thinking about a combination of those, really about how do you select the different algorithms and when to switch from one to the other?
Yeah, it's a great question. So there's a few factors that'll go into how you select your batch size. One of the most critical factors is kind of the turnaround time for experiments. And in this case, we kind of picked, you know, just a few days as our expected turnaround time. So we designed our batch size around that. If the turnaround time can be a lot longer, you might want to constrain your batch size a little bit so that you're getting feedback a little bit more quickly. But then you'll also want to factor in the relative complexity of your design space. If you've got a large number of variables that you're optimizing around, then you might want a slightly larger batch size so that your initial exploration phase of your optimization can capture some of that before it tries to go into the what's termed the exploitation phase of your optimization. So for switching from the weighted optimization to the Chimera optimization, for this case study, we did want to explore both. And so you see that we kind of did half and half, nearly half and half for weighted versus Chimera. And so that was done intentionally, mostly to get a better understanding of how this worked for that system.
Great. No, thanks a lot. It's a really interesting subject, and there's obviously a lot of discussion in the literature about how best to do this. So thanks for the question, Brittany. So a question here from Eric, really just asking about the automated LPPS system. What's the largest scale that you've managed to reach with that?
Yeah, so this is currently a development platform, and we use a 500 milliliter reactor for that. There's no inherent limitation for the size of that batch reactor. But as we've designed it and use it, it's a fixed volume system, typically delivering in the range of 20 to 40 grams of isolated peptide with protecting groups still installed out of the end of it.
Okay, great. And maybe just a follow-on question I have with that is kind of what's the next step after that scale in terms of to proceed with process development?
Yeah, so part of that is what our goal is when we're doing an individual run with the automated LPPS. So we use this with a lot of development projects for LPPS process development, and so within that context, it's used as our initial feasibility and familiarization with LPPS development. And so it'll reveal immediately whether there's some inherent solubility limitations that weren't captured by solubility prediction tools. But then we can take the crude product as well as IPCs collected throughout the process and do LCMS analysis to identify what some of the major impurities that are forming in the process are. Those can then be the target areas for process optimization. If you're making a 10 or 20 mer peptide in the liquid phase, you're talking about 20 to 40 different individual reactions, and you often need to be able to start with the worst performing reactions rather than doing your process optimization in too granular of a format. So it really provides a lot of detailed information to proceed into the more manual process optimization and facilitates that process.
Great, thanks. Question here from Adam about the HPLC platform. Can you give a sense of what platform and software you're using for that automated method development?
So this was internally, so for the optimization side of things, it's an internally implemented Bayesian optimizer that's an open source program. For a lot of the interface and how we interact with that, that's all internally developed software here at Snapdragon to do that type of development.
Okay, and maybe a follow-on question I have for you with that is what are the decision factors you use in terms of when to select Bayesian optimization versus other approaches like DOE for process optimization?
Yeah, so for broader and less well-defined design spaces, we find that things like Bayesian optimizers are going to be vastly superior to a DOE optimization. When you've got relatively constrained bounds for your process parameters, maybe plus or minus 5% or something along those lines, that's when we might reach for something like a DOE optimizer. We don't find that the approach for a DOE is going to be as robust when you're broadly exploring your design space as something like a Bayesian optimizer.
Great, thanks. And then just a question here from Jeffrey about the HPLC column optimization. Is temperature ever used as a variable for that optimization? In terms of variable temperature?
I'm sorry, can you restate the question?
Temperature ever used as a variable in that optimization?
Yes, we have. Not in this specific case study, but we have used that. And so, yeah, it's very flexible for the continuous variables that are being used for this. So anything basically that can be defined within the method can be utilized for that.
Okay, great. Yeah, I mean, the question that I have was, really some impressive uses of automation in the work you've done here. So can you maybe just sort of talk through some of the barriers of that people might find in terms of optimizing those things through automation? And what are you doing to address those kind of barriers?
Yeah, so I think I captured this to a small degree when I first went into the process optimization section of the webinar. And really the main barriers are how you piece together the different parts of technology. And so let me see if I can actually go back to that slide. And so one of the biggest pieces here is having flexible automation. And so what we've done over the years is develop a very modular and plug-and-play software platform that allows us to utilize a large variety of different types of hardware, kind of agnostic as far as the brand or equipment manufacturer for that. So being able to have this flexibility, as well as making sure that we understand when something's going wrong, is some of the biggest barriers to do this. And so, being relatively nimble and creative with how you implement these different types of technologies, I think is the biggest piece that's allowed us to kind of get over some of those barriers.
Okay, great, thank you. I think that's all the questions we have coming. I guess one final question for me: what's the really cool-looking photograph on the wall behind you? What is that we see there?
Yeah, so we love data here at Snapdragon and really love to understand reaction kinetics. So, relatively early in the life of our company, we wanted a very small scale, well temperature controlled reactor to monitor kinetics by React IR. So we have this piece of glassware custom built, and you can see the React IR probe kind of over here, I guess, through the center of it. Just going into this jacket-cooled small 25 mil reactor. And so it's just a photo that I took early on in my career of that reactor in use.
Great, well, thanks, Adrian. Okay, so just one extra question in here from Richard. So have there been combined Fmoc and Z synthesis examples? And did you ever try to perform the automated LPPS Z deprotection in flow? And with the did you ever try combining those together?
Yeah, so we have done hybrid fragment syntheses. The one example that comes to mind is when we had early on in the synthesis of a peptide functional groups that were quite sensitive to the basic conditions required for Fmoc deprotection. So we utilized CBZ chemistry for that. However, later on in the synthesis, we introduced a tryptophan residue, which we found to actually be quite sensitive under some of the conditions we're using to hydrogenation, where we're actually hydrogenating the indole ring. And so we switched from CBZ mid-fragment to Fmoc chemistry in order to accommodate the relative sensitivities of the individual portions of the sequence. So to date, we have not combined these within our automated LPPS platform. That's something we'll likely be aiming to do, over the next few months or so.
Okay. And then I just came in from Seshi, I think it is, and just asking, can we use this for small molecules? So I think maybe just this is for things like the automated liquid-liquid extraction. Is this just limited to complex synthetics, or is this something that you see being more broadly applicable?
Yeah, so there is ways to do this, and we've built a platform to do that for small molecules, largely focusing on automating execution of DOEs. It's a bit more difficult when you're trying to piece together a lot of different types of reactions. That's what makes this quite amenable to peptide synthesis and similar types of chemistries. Is it's very repetitive in nature. It's very it's very predictable on the types of reagents that you're using. But for an individual reaction that you want to repeat over and over again with variations on the reaction conditions, such as when you're doing a DOE, yes, it is very possible to do this with small molecules, and we have done this in the past.
Okay, great, thanks, yeah. I think that's all the questions we have. So, Adrian, thank you so much for a really interesting seminar. Thanks for answering the questions. And as you see here, he's left his contact information on the final slide here. So if you have any questions you want to ask Adrian directly, please email him there. And thank you very much for attending. Bye, everybody.